Yes, we recently published new work entitled Discrete Class 1 Molecules on Brain Endothelium Differentially Regulate Neuropathology and Experimental Cerebral Malaria. This work was performed in the lab of Dr. Aaron Johnson at the Mayo Clinic. Yes, let me start by giving a little background on the project. Many groups have addressed the role of immune cells in the development of cerebral malaria. It was ultimately discovered that CD4 T cells were found to be important early in infection, whereas CD8 T cells are required throughout the entirety of the disease. More recent work has established that CD8 T cells interact with brain vasculature in mice and in humans and infiltrate the brain during disease. The role of individual class 1 molecules on vasculature, however, has never been tested. After establishing relevance with human data, our figures 2 and 3 of the paper introduce our Cre-Lox system, which individually ablates either K of B or D of B under an endothelium-specific promoter. In these and supplementary figures, we show no perturbations of peripheral immunity at baseline or during infection. I would say the most important figures of the paper would be figures 4 through 8, since they hold the crux of the data. In figure 4, we performed intravital microscopy to evaluate T-cell dynamics. On the left, you can visualize T-cell interactions in this mouse that has K of B on vasculature. On the right, K of B has been ablated from vasculature. Quantified here, when K of B is ablated on vasculature, number of T-cells, velocity, and arrest were reduced. Adhesion molecule expression on endothelium was unchanged. Similar trends were seen with D of B ablation on brain endothelium during infection. Figure 5 focuses on immune changes in the brain during infection. With K of B ablation, the numbers of CD8s infiltrating brain is reduced, but more CD4s are producing interferon gamma. When comparing brain, meninges, and cervical lymph node, only brain had a significant reduction in antigen-experienced CD8 infiltration. With D of B ablation, numbers of overall CD8s did not change, but their interferon gamma production was still lost. As with K of B, only brain had significant reduction of antigen-experienced CD8s. In a neurotropic virus model, we showed that endothelium can present antigen from within the brain as well, and that when either class 1 is lost, antigen-specific CD8 entry is lost as well, just as in malaria. In figure 6, we evaluate vascular leakage in the brain during infection. Loss of either K of B or D of B results in complete protection from vascular leakage, with K of B only and D of B only Cre negatives having intermediate leakage phenotypes, with D of B being more severe. To confirm that these changes are due to CNS-specific endothelial ablation, we perform parabiosis to equalize the peripheral immune environment and reduce any off-target effects. We perform dual gadolinium-enhanced MRI on the parabionts, as well as subsequently physialbumin assay, and were able to confirm the findings in our previous results. In figure 8, we examined RNA transcriptomics data and were able to pinpoint key changes in the neuroinflammatory profiles of the brains dependent upon the class 1 expression on endothelium. On day 6 of infection, we examined cell death and saw no change with loss of K of B. With D of B, however, we saw cell death of endothelium and neurons when D of B was present. K of B Cre negatives have a lower average clinical score on day 6 than D of B Cre negatives, which may explain this finding. When allowed to progress to endpoint, expression of granzyme B messenger RNA was significantly reduced with loss of K of B or D of B, which correlated with the activation of genes in the neuronal cell death pathway and with survival of the animal. Novel findings are H2K of B and H2D of B are important for adhesion at the brain vasculature. Loss of K of B stops CD8 infiltration, but D of B does not. Loss of either results in reduction of antigen-experienced CD8s. CD8 interferon gamma production is dependent upon class 1 interaction. Vascular leakage is dependent on class 1. K of B and D of B differentially modulate cell death and disease progression, and ablation of either protects against the development of ECM resulting in survival. Our data demonstrates that these molecules coordinate activation, entry, and induction of neuropathology. Their modulation may therefore hold therapeutic benefit in cerebral malaria. Thank you for your interest in our work and enjoy the paper.